Well, this is the third part of our lecture series on the physical structure of DNA. And in the last one, we talked about how Watson and Crick, using the data that they had from Rosalind Franklin as a template, were able to put together the final physical structure of DNA. And probably this part of the lecture is the most important thing to you. It's one you may probably come back to, because we're going to talk about, lay out exactly what the Watson and Crick model said. And this is important because for working problems, which we're going to do in a later lecture, another part here in a moment, um, as well as from the book and exams, it's really some of the information that you'll need will be right here. So just to kind of refresh us where we were, Watson and Crick decided that we have a helical model. That model, based on Rosalind Franklin's data, is about 20, this is angstroms, but we're talking about 2 nanometers wide. The steps are fairly regular, and they're about 0.34 nanometers or 34 angstroms tall. And what's very important is what Watson and Crick came up with was the fact that this is an anti-parallel model. It's double-stranded DNA, which is the most stable biological form of DNA, has one strand going 3' 5 and the other going 3' 5, but in the opposite direction. Okay, so this allows it to maintain a regular structure and form. Now, taking all this, Watson and Crick looked at their model and they came up with some basic structural pieces of information that are going to be important to us. Okay. The model they were looking at turns out to be the B form. We'll talk about some alternate forms in the next little short lecture section. But for right now, the Watson-Crick model is what's known as the beta or B form. Okay. Now, what they say is that we've got 2 polynucleotide chains, right? They are coiled around each other, which making double-stranded DNA, which forms a helix. Okay, they tend to be what's known as right-handed. Right-handed means that if you were to grab the top and twist it clockwise, it would screw into the ground or wherever you're trying to go. Okay, left-handed is that you'd have to turn it the other way to make it screw in. Okay, so these are right-handed helix. They go like this. I actually had redid an old Willis wagon, and this is from the 1940s, and back then they believed that if on which side the lug nuts were on one side or the other, needed to go in opposite directions. So on one side, with the wheels going this way, you had right-handed, which always means they always stay tight when it was going forward. The other side, they would make them left-handed so that you were always getting tightened as you went forward. At least that was the concept. Of course, modern cars are pretty much all right-handed, but it was an interesting thing to have to take this car in to have anything done. Have to remind these people that no, it's left-handed on one side and right on the other, because if you didn't, they would always mess it up. This is a little side trip for some reason to kind of keep you in your mind what right-handed and left-handed mean. All right? Okay. As we said, both chains then have to be anti-parallel, right? So instead of this way, one goes this way. That was a major discovery. And the bases are complementary, okay? So A to T, G to C as we work through it. The bases themselves then are relatively flat. They're slightly torqued, but they're relatively flat. They're perpendicular to the central axis, and they're stacked as far as about 0.34 nanometers apart, okay? So they're somewhere like this, and they're fairly regular. Now, they do this as we move up, okay, but they're stacked relatively close. So they're just slightly turned as we go from one step to the other. I think probably the easiest thing to do is think about it as a spiral staircase, right? And the spiral staircase, you go up, each step is slightly turned so that you move forward up, okay? And this is exactly true here. With each of these little slight turns, okay, it takes about 10 bases for you to go around. So if I were to sit here and start up the stairs, taking one step at a time, go all the way around, swivel around, I'd be back facing you after 10 steps. Okay, that's the basic idea. It turns out, and we know it's slightly more than that at this point, maybe closer to 11, but that's all right. For our purposes, 10 steps will work. Now, this is important in both of these pieces here. This 10-step and this distance are important because genetics problems, we could ask you, okay, I have a gene, it's approximately 8,000 base pairs in length. How many turns of DNA were there? Okay, just divide by 10. How long is that piece of DNA? Okay, we'll just multiply by 0.34 nanometers. So 8,000 times 0.34 nanometers tell you the approximate distance. That's all you need to work these fairly straightforward problems, okay? Again, just the bases on the opposite strands are bonded to one another with hydrogen bonds, 2 for AT, 3 for GC, making the GC region slightly more stable. And the backbone is linked together by phosphodiester bonds. Now, this is going to be really important because what you have here is you have these bases, okay, hooked together by 2 bonds or 3 bonds, bases, 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 bases. The center part of the DNA molecule is essentially Velcro, if you want to think of it that way. It's you have these little hydrogen bonds, fairly low energy bonds, easy to break. Okay, but if you've got millions of them, which you do, certainly hundreds of thousands in a chromosome, you're going to have a fairly strong structures like Velcro, okay? But any one of those can pop apart. So if I need to get in here and read this piece of code inside the DNA, it's very easy to do that. I can pull apart the DNA, read this part, and then these little hydrogen bonds will form back together. As a whole structure, it's very stable and strong, like a piece of Velcro. You know, if you just grab it and try to pull it, it's not. But individually, a part of it you can pull apart. The other is these phosphodiester bonds that form the backbone like this. These are very flexible, so they allow that torquing that you need to make it a helix. Okay, they're very strong and stable bonds. In fact, as I've said before, DNA can be found that's thousands of years old, can be extracted because these bonds are still holding the thing together. They're highly flexible, yet they're very strong phosphodiester bonds. So you get both things that you need. These little hydrogen bonds are allowed to pull it apart, so you can get to it, but the backbone is very strong and stable and flexible. Okay? But having all this, you end up with what are known as major and minor grooves. This is the minor groove here, it's a major groove here. That's true. If you want to see it, just take a fairly large piece of rubber band or piece of string, whatever, put your fingers and just keep twisting it around, and you'll see that it will form where the 2 band parts come together, will form minor grooves, and then where the other parts come together, you'll have what are known as major grooves, okay? And then finally, the helix itself, the distance here remains relatively constant, about 2 nanometers apart, okay? So about 2 is one of the more stable things that we see for the system, all right? Now, that's our model. Okay, again, these are angstroms instead of nanometers, but it doesn't matter. It's a nice consistent 5. You've got minor grooves, major grooves, you've got base pairing and all of that, okay? So then, okay, Watson and Crick then had to say, how does this work as a genetic material? Remember, there were 2 questions that we started this whole section out on. We talked about how does it all fit together? How do you take nucleotides and make DNA, this structure? And then how do you make it into a successful genetic material? Well, remember that we said there are really 4 basic things that you need. And this may vary from the book, okay? Don't too worried about it. These are what I'd like for you to know, and I think these are somehow fit together. Even the book might just mention 3 of them. Okay, replication. We've got to be able to effectively transmit the information to the progeny as well as make the phenotype when we get there, store lots of data, and have a basis for mutation. Watson and Crick, in their little small Nature paper that they proposed, the model addressed these. In particular, they said that replication was probably semi-conservative. That's going to be really important when we talk about replication. Okay, semi-conservative means that we're going to pull these pieces apart. Okay, so the pieces being the double-stranded DNA, and we're going to make a new copy over here and a new copy over here, and that's what we have. So we always end up with one strand of the DNA being old and one strand, the other side, being new. Same thing happens over here. That's semi-conservative. We'll talk more about that. But they propose that model, and it becomes important. The other thing that is really important here is information storage. Right? Think about it. You've got 4 bases, A, T, G, C, which means that for any gene region, you're talking about N, the number of bases, to the 4th power is how much possible information you could have. So let's take a little tiny section here. Let's just take 1, 2, 3, 4, 5, 6, 7, 50 bases making up a region. If you take that to the 4th power, there are over 6 million possible combinations there. It could be A, T, or A, G, or A, G, 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 A. All the possibilities, there's over 6 million. Well, think about, the smallest genes you see, even mitochondrial genes are 1,200 bases. Many genes can go up, the average is over 5,000 to 6,000 bases for a given gene in most systems, and they can go to over 100,000 bases. So think about all the possibilities that you have, right? Every one of those bases could change. One of those changes is a mutation, right? So just a small section of DNA has just an infinite number, almost, of possibilities for that. In fact, you take just a small section of thing, and you could have, it's more storage capability than any computer, all the computers ever put together that we have so far on this planet. Storage in just the DNA that's probably in one cell in your body, that 1-2 meter, that's 6 feet of DNA that you tend to have. Okay. All right, so now we have all the structure, and I think you should go back and really look at this page and know all this information and work through it. The next little model, what I want to do is I want to just briefly go over the other sides besides the beta form, a couple of things that just to let you know. And then in the last lecture on this series, we'll talk about some problems. Come and actually work a few simple problems based on primarily this information.